In a previous video, we introduced a group of cellular organelles that were related through their participation in the endomembrane system. In this video, we will introduce the remaining cellular organelles, including peroxisomes, mitochondria, and chloroplasts. Although they are grouped together by their exclusion from the endomembrane system, they have other noteworthy similarities, including their ability to metabolize oxygen in one form or another, and their proposed extracellular origins. This latter characteristic has led them to be called the semi-autonomous organelles, meaning that at one time they were independent from the cell. Our first organelle is the peroxisome. The peroxisome is a spherical organelle that under an electron microscope appears to have a single membrane surrounding it and a dark crystalline core near its center. The name peroxisome is derived from the significant presence of hydrogen peroxide, which is both made and broken down in the peroxisome, as shown by these two equations. Enzymes in the peroxisome pull hydrogen atoms from organic molecules, shown here as the letter R, and add them to oxygen to create hydrogen peroxide. Still other enzymes continue the process by taking more hydrogen atoms from other organic molecules and adding them to hydrogen peroxide to create water. This type of reaction is called a reduction-oxidation reaction, and it is a specialty of the peroxisome, just like hydrolysis reactions are a specialty of the lysosome. Redox reactions are used in the peroxisome to carry out its special functions, which include creating fatty acids and breaking down drugs and other toxins. These compounds serve as a source of hydrogen atoms to make hydrogen peroxide. The role of peroxisomes in detoxification and preventing damage from oxygen causes some people to describe it as a first aid center inside the cell. Our next organelle is the mitochondria. Mitochondria are also not part of the endomembrane system. They are often considered the power plant of the cell because they are the site where the cellular fuel source, ATP, is created. Unlike most organelles, mitochondria have two membranes, an outer membrane shown here in orange and an inner membrane shown in red. The inner membrane is highly folded to increase its surface area. Individual folds in the inner membrane are called cristae. Within the mitochondria, two spaces are created. The light orange space between the outer and inner membrane is called the intermembrane space, for obvious reasons. The space inside the inner membrane is called the matrix, and it is where most of the mitochondrial operations are carried out. The mitochondria make ATP through the process of cell respiration. This process not only makes ATP, but also uses the oxygen that we breathe in and produces the carbon dioxide that we breathe out. It begins when carbon compounds, like sugar, enter into the matrix. A series of chemical reactions called the Krebs cycle strip electrons from carbon compounds and make carbon dioxide. The stripped electrons are shuttled from the Krebs cycle to a series of proteins along the inner membrane called the electron transport chain. Oxygen enters the mitochondria and pulls stripped electrons through the electron transport chain. When added to oxygen, the extra electrons create water. The energy from the moving electrons is used to make ATP. We will explore a lot more about these two important processes, the electron transport chain and the synthesis of ATP, in a future tutorial. Another important but unusual resident of the matrix is the mitochondrial DNA. The DNA inside the mitochondrial matrix is circular and much smaller in size than the linear chromosomes found in the nucleus of the cell. In fact, it closely resembles bacterial DNA. The similarities between mitochondrial and bacterial DNA have led some scientists to propose a prokaryotic origin for the mitochondria. This will be explained more in a moment. This slide is a good place to pause and review some of the important terms associated with the mitochondria. On the left are structural terms that identify the different membranes and spaces within the mitochondria. Starting on the outside of the mitochondria and moving in, we encounter the outer membrane, the inner membrane space, the inner membrane with its multiple cristae, and finally, the matrix. On the right are some of the important functional terms for the mitochondria. It is the site of cellular respiration and ATP production in the cell. Oxygen is consumed and carbon dioxide is produced. Two important processes in cellular respiration are the Krebs cycle, also known as the citric acid cycle, and the electron transport chain. The mitochondria also houses its own circular DNA. It may be useful to pause the video and point out to yourself the different structures and images associated with different functions on the unlabeled mitochondria shown here in the middle. The chloroplast as an organelle is only found in plants where it functions as the photosynthesis center of the cell. Photosynthesis is the reverse of cell respiration. It uses energy from sunlight to convert water and carbon dioxide into oxygen and simple sugars. Like the mitochondria, the chloroplast has several different membranes. The outer membrane and inner membrane are shown here in sea blue and light green. The space in between the two membranes is called the inner membrane space, shown here in light blue. A third membrane, called the thylakoid membrane, is functionally the most important membrane. It is where the light-capturing proteins and pigments for photosynthesis reside. It has lots of folds in it to increase its surface area and make room for more proteins. Under a microscope, these folds form interconnected sac-like compartments called thylakoids. A stack of thylakoids is called a granum. The space inside the thylakoid is called the thylakoid space. The space outside the thylakoid is called the stroma, shown here in light green. It is where the enzymes and intermediates of the Calvin cycle are found. In photosynthesis, the Calvin cycle is responsible for taking carbon dioxide from air and turning it into sugars that the plant can use for energy and building blocks. Like mitochondria, chloroplasts have their own circular DNA, and it is located in the stroma. Because it houses important chemical pathways and circular DNA, the stroma is often compared to the mitochondrial matrix. This slide is a good place to pause and review some of the important terms associated with chloroplast. On the left are the structural terms that identify the different membranes and spaces within the chloroplast. As you look over them, you may want to point at the structures they refer to in the provided image. We start on the outside of the chloroplast with the outer membrane, followed by the inner membrane space, the inner membrane, and the stroma. Inside the stroma we have the thylakoid membrane that surrounds the thylakoid space. A stack of thylakoids is referred to as a granum. On the right are some of the important functional terms for chloroplast. It is the site of photosynthesis in which energy is captured from the sun and then used in the Calvin cycle with carbon dioxide to make simple sugars. Like mitochondria, the chloroplast has its own DNA. The following section provides an explanation of the prokaryotic symbiont theory, which describes what many scientists think to be the origin of both mitochondria and chloroplasts inside the cell. A prokaryotic origin for peroxisomes is also proposed by some, though it has less evidence. Long ago, when cellular life was first developing, scientists think the atmosphere had a different composition than it does today. Oxygen, shown here in red, was present in lower amounts, and it generally was considered to be toxic to cellular life, causing the gradual breakdown of proteins and nucleic acids by oxidation. It is believed that the peroxisome, shown in blue, evolved as a primitive organelle to absorb oxygen and chemically modify it, first to hydrogen peroxide and finally to water, which could then be released harmlessly into the cell. This gave the first eukaryotic cells a way of dealing with the toxicity of oxygen. For energy, some cells would scavenge from the environment, like this blue prokaryotic cell. Larger eukaryotic cells would gain energy by preying on these smaller cells through a process of phagocytosis, in which they would extend their membrane around their prey, encasing them in a new compartment, where they would then be digested. And this was life for millions of years, until two very unusual things happened. The first was the arrival of a new type of prokaryote that carried a primitive electron transport chain and ATP synthase. With this new molecular machinery, the prokaryote was able to use oxygen to power the electron transport chain and drive ATP synthesis and convert the oxygen to harmless water in the process. This new mechanism gave the prokaryote an energetic advantage and allowed it to thrive over its neighbors. The second event was both common and at the same time unusual. The common part was that, like all other prokaryotes in the primordial soup, our ATP innovator got swallowed up by its larger eukaryotic cousin. The unusual part was that once it had been swallowed, for some reason, it stayed inside its encasing membrane and wasn't digested. So, it just kept using oxygen and making ATP, like it was programmed to do. The larger predator found itself with a new and abundant source of ATP, and the prokaryote found that the eukaryotic cytosol was a much more plentiful source of nutrients and kept it protected from other cells. As a result, its ATP production actually increased, allowing it to supply the needs of both itself and its larger host cell. When the eukaryotic cell divided, the prokaryote in its protective membrane also divided, and each new daughter cell received its own prokaryote symbiont. A symbiont is an organism that lives on or in another organism. In this case, the host and symbiont working together created a much better organism that soon outpaced all its competitors and eventually led to all other eukaryotic life forms today. We don't exactly know if this is the way the first mitochondria evolved, but there are some pieces of evidence to support it. For example, mitochondria and chloroplasts have their own DNA and their own ribosomes. In mitochondria, these are found in the matrix, and in chloroplasts, they reside in the stroma. Both compartments correspond to the cytosol of the ancient prokaryotic ancestor. Interestingly, the mitochondrial versions of DNA and ribosomes more closely resemble prokaryotic DNA and ribosomes than eukaryotic versions. Finally, we also know that mitochondria divide within the cell in a process that mimics binary fission, a distinctly prokaryotic process. This concludes our survey of the semiautonomous organelles, including peroxisomes, mitochondria, and chloroplasts. This video is a product of the Biology Education Animation Research Project at Brigham Young University, Idaho. Video animation, narration, and production were carried out by Dr. Stephen Christensen. If you have comments, questions, or would like to help make additional video tutorials, please contact Dr. Christensen at the provided email. In this video, we will introduce the structures of the cytoskeleton. The three principal fibers of the cytoskeleton are microtubules, actin filaments, and intermediate filaments, and each will be presented with information on their structure, properties, functions, and locations inside the cell. As humans, our skeleton form an important part of our anatomy. Without them, we would be a formless blob of skin and muscle heaped on the floor. Our skeleton also plays a big role in our mobility. Even if we remove the pain associated with a broken leg, it would still be impossible to walk or move with it in a broken, unsupported condition. Likewise, each individual cell relies on a cytoskeleton for its shape, integrity, and mobility. Over the next several minutes, we will introduce you to the three fiber types that make up the cytoskeleton. These include microtubules, actin filaments, and intermediate filaments. We will also describe the primary roles each play in maintaining cell structure. Later sections will dive deeper into the structure and additional functions of each group. The largest of the cytoskeleton filaments are the microtubules. They have a diameter of roughly 25 nanometers. In this tutorial, we will use green to represent microtubules. Microtubules are shaped like a steel pipe with a hollow core, and like a pipe, they are fairly rigid and inflexible. This rigidity is essential to the role they play in helping the cell resist compression. In the cell, microtubules always grow out from a central point near the middle of the cell called the centrosome. In the center of the centrosome is a well-known cellular structure called the centrioles. As the microtubules spread out from the centrosome, they weave around the organelles like the nucleus and push up against the walls of the plasma membrane. In this way, microtubules help the cell resist compression or pushing forces that would otherwise squish it flat. Actin filaments are the smallest type of fiber in the cytoskeleton, with a diameter of only about 6 nanometers. Actin filaments are also sometimes called microfilaments. They have a rope-like structure that is very flexible, meaning it can bend much better than microtubules, but it is inelastic, meaning it doesn't stretch well. We will use red to depict actin filaments throughout this video. In the cell, one of the primary functions of actin filaments is to form a protective netting all around the plasma membrane. This netting is called the cell cortex, and it helps protect against the pulling forces that might rip the cell apart. The purple dots in this image represent complexes of integral membrane proteins that anchor into the plasma membrane and form hubs for connecting the ends of individual actin filaments. Our final set of filaments are called intermediate filaments. At an average diameter of 10 nanometers, they are in between the sizes of microtubules and actin filaments. Intermediate filaments are made from wrapping over 30 separate strands together into a cable-like fiber. Like steel cables, they have a very high tensile strength, meaning that they are difficult to break, but they remain flexible and can move side to side. Unlike steel cable, they are also elastic, meaning they can be stretched and when released fall back to their normal shape. In these videos, our intermediate filaments will be represented in blue. In the cell, intermediate filaments are particularly concentrated around button-like structures called desmosomes. Desmosomes anchor a special type of integral membrane protein that connects one cell to another. Intermediate filaments also extend across the cell, connecting desmosomes on one side to those on the other, and creating a strong fibrous network that keeps the cell locked into other cells around it and resists tension forces that might pull the cell apart. Now that we have surveyed the cytoskeleton in general and introduced its primary components, let's take a closer look at the structure and functions of each filament, including the important role played by motor proteins. We will start with microtubules. Now it's time to take a closer look at the structure of microtubules. Microtubules are made from assembling smaller protein subunits together into a long fiber. The smaller subunits are a peanut-shaped protein called tubulin. The tubulin is arranged head to tail in a series of 13 strands that wrap around to form the hollow tube structure of the microtubules. Microtubules are directional, meaning that they have a front and a back, as shown by the dark and light shades of green in this illustration. One end of the microtubule is called the plus end, because subunits are added more quickly at this end. The other end adds more slowly or sometimes even loses subunits, and is therefore called the minus end. Some beginning students misinterpret these names as an indication of positive or negative charge at the opposite ends. This is incorrect. Neither end carries any significant charge. In the cell, the minus end is always located near the centrosome, while the plus ends are located distal to the centrosome near the plasma membrane. Inside the cell, microtubules are dynamic, meaning that a single microtubule grows by adding tubulin subunits or shrinks by getting rid of tubulin subunits. The ability to add or remove subunits allows the cell to easily disassemble microtubules in one location and reassemble them in a new location where they are more needed, similar to the scaffolding platforms that construction companies use when making large buildings. As we have seen previously, microtubules are typically spread across the cytoplasm, where they function in resisting compression forces. In this arrangement, they also create a type of intracellular highway system that is used to shuttle transport vesicles between organelles. The microtubule network is also critical to maintaining the structure and organization of large organelles, like the endoplasmic reticulum, because if the microtubules are disrupted or broken, these organelles quickly fade from view. During mitosis, the microtubules form a special structure called the spindle apparatus, where two pairs of centrioles separate to opposite ends of the cell and then grow overlapping microtubules between them. The condensed chromosomes are contacted by still other microtubules, which grow out and contact them at the centromeres. These microtubules then shrink back, pulling the two halves of the chromosomes to opposite sides of the cell. Flagella and cilia are two additional structures that are made of microtubules. Flagella are long whip-like tails that are used by cells for propulsion, much like a propeller on a motorboat. Cilia are similar to flagella, but they are generally much smaller and much more plentiful. To propel the cell forward, the cilia beat in unison like the oars of a Viking longboat. In animals, cilia line the cells of the trachea and lungs, and their beating movement carries mucus and other cellular debris out of the respiratory tract. If we could look inside a cilium or flagellum, we would see 9 microtubule doublets arranged in a circular pattern. Dozens of small motor proteins are attached to each filament in the bundle. When these motor proteins start to move along neighboring filaments, they cause the filaments to slide relative to each other, making the cilia or flagella bend, first in one direction, then in another. Motor proteins are an essential component of each of the previous examples. Motor proteins are able to bind and move along microtubule filaments. Microtubules have two distinct types of motor proteins, kinesins and dyneins. Kinesin motors move along the microtubules toward the plus direction. They have two motor heads that alternate moving forward along the filament in a step-like fashion. Kinesins play an important role in the formation of the spindle apparatus and vesicle transport. Dynein motors move toward the minus end of microtubules. Unlike kinesin, they seem to have a more rotary movement that makes short casts of a flexible chain, which is then used to pull the motor unit forward. Dyneins play an important role in moving chromosomes toward the opposite sides of the cell in cell division, and in the beating action of cilia and flagella. We will now move on to our second cytoskeleton component, actin filaments. As before, we will take a closer look at the structure of actin and its assembly and disassembly. We will then identify several important locations and functions of actin filaments and their associated motor protein, myosin. Actin filaments have a helical shape created from two identical strands that wrap around each other. Each strand is made by arranging smaller protein subunits of globular actin, or G-actin for short, that assemble in a head-to-tail fashion at either end of the filament. Each end of the actin is different, making it a directional filament. Like microtubules, one end is able to assemble subunits faster than the other, so it is called the plus end. The opposite end with slower assembly is called the minus end. Inside the cell, actin filaments are also dynamic, meaning they grow and shrink, allowing easier assembly and disassembly where they are needed in the cell. We have already mentioned the primary role that actin filaments play in creating the cell cortex. Actin filaments are also involved in several other key structures inside cells. The most well-known site of actin function is in muscle fibers. Actin filaments are arranged in opposing bundles with bundles of myosin motor proteins between them. During contraction, the myosin bundles pull opposing actin strands toward each other. Actin filaments are also important in cell division, where they form a contracting circular band of fibers called a contractile ring. Myosin motors interspaced between these fibers cause the fibers to constrict, causing a cleavage fur to form that ultimately divides the cytoplasm of the cell in half. Some cells undergo the process of phagocytosis. Actin filaments near the cell cortex grow outward, extending the plasma membrane out and around nearby bacterial cell. Many of actin's associated functions depend on the ability of myosin motor proteins to move along the actin filament. Generally, myosins move in the plus direction. A single myosin step involves the extension of a motor head, shown here in light blue, binding to the filament, contraction, and subsequent release of the filament. This cycle is repeated over and over. Intermediate filaments are our final component of the cytoskeleton. As with our previous filaments, we will take a brief tour of their structure and then discuss some unique properties and functions. Like other cytoskeleton components, intermediate fibers are assembled from much smaller protein subunits. While actin and microtubule filaments are made from sphere-like subunits, the subunits of intermediate filaments are more column-shaped and form a tetramer, meaning that it is made from 4 smaller subunits. These tetramer subunits arrange themselves into 8 long strands that wrap around each other to form a very strong but flexible cable. This cross-section view of a filament shows the 8 strands that make up the filament and the 4 subunit coils that make each strand. Intermediate filaments don't really have a distinct front and back, so they are not directional. And even though they do assemble and disassemble, they don't do it very often, so they are considered much more stable than the other filaments. We have previously discussed the role of intermediate filaments in holding cells together and keeping them from being torn apart. Intermediate filaments, called neurofilaments, also line the long axon processes of nerve cells, keeping them from breaking. Another whole family of intermediate filaments, called keratins, are the primary components in claws, scale, skin, hooves, and horns. The keratins themselves are very diverse, with over 30 different genes encoding the keratin proteins. A final example of intermediate filaments is found in the nucleus of all cells. If you could open up the nucleus, you would find a class of intermediate filaments called lamins. The lamin name is derived from their formation of the nuclear lamina, a crisscrossing pattern of lamin filaments located on the inside of the nuclear envelope that gives the nucleus its shape and rigidity. This video is a product of the Biology Education Animation Research Project at Brigham Young University, Idaho. Video animation, narration, and production were carried out by Dr. Stephen Christensen. If you have comments, questions, or would like to help make additional video tutorials, please contact Dr. Christensen at the provided email.
Did it ask permission?